To talk about endosymbiotic theory, and in particular the evidence that mitochondria came from or arose from some free-living cells that were engulfed by eukaryotes. So in this image, we see something similar to what we saw with the tree of life. However, we're seeing something different, in particular, this horizontal line. When we looked at phylogenetic trees and cladograms previously, we didn't see any of these horizontal lines going from one branch to another unless those branches were diverging, in cladograms specifically. Here we're seeing this horizontal line, which is trying to describe that an organism was actually engulfed by another organism and became part of that second organism. So we see two of those lines, one here and one here, and these represent what we think was the engulfing of a bacterium, which became the mitochondria, and a bacterium, or cyanobacterium in particular, that became the chloroplast. And we see these lines, the horizontal lines in particular, going from bacteria to the base of the eukaryotic branch. And so that we refer to this sometimes as a proto-eukaryote. It just means that it's an ancient eukaryote that wasn't quite like what we know eukaryotes to be today. And this is when we think the free-living organism was engulfed and eventually became the mitochondria. And then we also see a transfer here of cyanobacterium, or really more of an engulfment rather than a transfer. And this happened at the base of the plant and algae branches here, because both of those contain very similar chloroplasts. And we see that that happens at the base of this branch, but it does not include the animal and fungi branches. So what kind of evidence is there for this event? Is it just coincidental that the mitochondria and chloroplasts happen to have the same kind of shape as free-living bacteria? Or is it more than that? Well, it's actually a lot more than that. And in particular, if we look at mitochondria, mitochondria have that shape of a bacterium. So they're rod-shaped, and that's one particular shape of bacteria. And they also have a double membrane. And the compositions of the two membranes are different. So what the theory is that the inner membrane was from the original membrane of the organism, and the outer membrane is from the membrane of the organism which engulfed it. So the inner membrane is more similar to bacterial membranes. The outer is more similar to eukaryotic membranes, or proto-eukaryotic membranes, I guess you could say. So mitochondria, as we know, are the site of energy metabolism. So we talked about this in the first bio, and we talked about the TCA, or tricarboxylic acid cycle, also known as the Krebs cycle. And in this process, this is where the carbon skeleton from glucose comes in, and it is oxidized and eventually released as carbon dioxide, or all those carbons in glucose are released as carbon dioxide. And eventually we get these molecules of NADH as well as FADH2, and they're used in the mitochondrial membrane by enzymes which create a proton gradient, and then ATP synthase is an enzyme which uses that proton gradient to actually make ATP. And so that's where most of the ATP comes from in our cells. And this is a process that is also used by plants and plant cells as well as algae. Now, you don't need to know all of that process because we're not going to go into the enzymes and the different steps of that. But what you should know is that the mitochondria can generate energy. And actually, if you take a mitochondria out of a eukaryotic cell, it can continue to generate energy if you provide it with the correct substrates. So it won't survive forever, but it'll survive for a number of hours, a fairly long time, in terms of taking, a, a part of a cell out of its normal environment and allowing it to survive. So the mitochondria is pretty independent. It doesn't need a lot of things from the host cell. And in fact, you might have learned when you're talking about mitosis that mitochondria don't divide in the same time frame as or in conjunction with the host cell. Mitochondria go through their own replication cycle. They have their own DNA, which governs this. So they copy their own DNA at the appropriate time, and then they actually split and go through a doubling mechanism, which is actually also very similar to the doubling mechanism of bacteria. So those are some more reasons why we think mitochondria originated as bacteria. Now, obviously, this isn't something that can happen just whenever a bacterial cell is engulfed by another organism. So there are lots of eukaryotic organisms that will engulf bacterial cells or consume them, and take them apart, break them down, use them for energy. But we believe that this happened at least once. So I just mentioned that DNA that mitochondria carry around, and that if we look at the sequence of that DNA, it's even more evidence that this structure in eukaryotic cells was or has an ancestor which was once a free-living cell. So if we look at this cladogram here, this mitochondrial DNA sequence is most similar to this typhus bacterium DNA. So it's very similar to this DNA. The mitochondrial DNA is much smaller than a bacterial genome, so we think that the mitochondria probably lost a lot of genes because it wasn't using the genes that it needed to live in the environment and deal with a changing environment inside of the cell. It's probably pretty consistent. And then similarly, if we compare the DNA found in chloroplasts, which work similar to mitochondria in that they also have their own replication schedule or cycle, and they have their own DNA, which they copy themselves. This DNA in chloroplast is very similar to cyanobacterial DNA, meaning that it seems that the original cell, which was engulfed, which eventually became a chloroplast, was most likely related to an ancestor of present-day cyanobacteria, which are bacteria that photosynthesize. And so this is really interesting, and again, it suggests that these events happened at least once and evolved or progressed into a mutually beneficial relationship so that the larger engulfing cell, the proto-eukaryotic cell, then maintained these inner cells, essentially, which became the mitochondria and the chloroplasts. But these plastids, or the mitochondria and the chloroplasts, still retain independent characteristics, including their own DNA, the ability to metabolize energy, they make their own enzymes, and they replicate on their own schedule. So a question that this brings up is, how do we kind of process this? How do we think about this in light of genes? So do we include the mitochondrial genome in the human genome? And if we say yes to that, then we need to start thinking about not just entire cells being engulfed, but just the exchange of DNA from organisms, and maybe including that in a phylogeny. So this is, I forget what they call it. It's I think, like a vine of life instead of a tree of life or something like that. And so it's supposed to give you the idea that yes, we know about these two events, bacteria being engulfed that gave rise to chloroplast, and the bacteria that gave rise to mitochondria. We know about these, but these other horizontal lines are suggesting that there are lots of transfers of DNA, not necessarily engulfment of cells, but the transfer of DNA from one organism to another that other organism eventually ends up using, and it maintains and retains those genes. Now, most people would not agree that this is a useful approach to phylogeny because this just looks like a mess, right? It's very difficult to look at this. It's also not well accepted because it is difficult to figure out which organism the gene might have originated in. So who's giving who a gene here between these two organisms? Did the gene originate in the Korean archaeota or the eukaryotic archaeota? And usually we instead will just include that gene separately and build our phylogeny accepting that some genes that we use in most organisms are going to have originated in other organisms. But if you look at the overall scope, maybe 98% of your genome comes from just your organism, and then maybe 2% comes from genes that were acquired from other organisms. So it's definitely interesting, but it might not be super informative, and those genes are not as significant on a large scale, or no one of those genes is as significant on a worldwide scale as these two events here, which is the engulfment of the entire organism.